Okay, let's go ahead and continue talking about those GPCR receptors. This G protein coupled receptor right here, its pathway is known as the G protein coupled receptor phospholipase C. Phospholipase C is going to act as the amplifier enzyme. So to begin this signal transduction pathway, we have to start off with a ligand, and that ligand needs to fit into that receptor. Once that ligand binds to that receptor, the G protein gets activated. After the G protein gets activated, it's going to go ahead and activate that amplifier enzyme. And we said before that an amplifier enzyme is an enzyme that's going to activate some other molecules, and those other molecules are going to activate some other molecules. So basically, you amplify the response that you get at the end. So the amplifier enzyme right here is an enzyme known as phospholipase C. What it does is it takes some of the membrane phospholipids within the cell membrane and it converts it into two compounds. One is known as DAG, which stands for diacylglycerol, and it will also convert some of the membrane phospholipids into IP3 or inositol triphosphate. Now, diacylglycerol stays within the cell membrane and it will go ahead and activate another enzyme, protein kinase C. As we said, whenever you see the term kinase, you know that this enzyme is going to go ahead and phosphorylate a bunch of proteins. Once those proteins are phosphorylated, then the final cell response that we are after could go ahead and take place. Inositol triphosphate is going to exit the cell membrane, enter into the fluid inside of the cell, and it will go and bind to a special receptor found on the surface of the organelle endoplasmic reticulum. Once it binds, the calcium that's stored inside of the endoplasmic reticulum will go ahead and get released. Once calcium is released, calcium is going to go ahead and bind to some other proteins, or calcium can cause some vesicles to release the chemicals that they are carrying. So at the end, you also get a different cell response. Now, what's an example of a ligand that binds to this G protein coupled receptor and activates the phospholipase C enzyme? And this cascade of events can go ahead and take place. An example is angiotensin. So we have a hormone known as angiotensin 2, and angiotensin 2 causes your blood pressure to increase. And angiotensin 2 binds basically to that receptor right here, activating this signal transduction pathway right there. This image right here shows you that you can have one receptor, and that one receptor can actually bind to multiple ligands. Not at the same time though. So if we take a look at this image right here, here's the receptor, and here is one ligand binding to it. And here's the same receptor, but yet there's a different ligand binding to it. How come those two different ligands can bind to the same receptor? As you can see, they have the proper shape that would allow them to fit within that receptor. Now, those two ligands are not going to bind at the same time though. They're actually going to compete. So they're going to compete against each other, and whoever gets to the receptor first will go ahead and bind to it. Those two ligands, if they are going to activate the receptor and lead to a response, we call them an agonist. So we call them agonists to each other. An example of two ligands that are agonists to each other and can lead to the same response, epinephrine and norepinephrine. So epinephrine and norepinephrine, they would lead to the same response. We usually produce them when we are stressed out. So they are responsible for your fight or flight response. And as I said, epinephrine and norepinephrine, they would usually compete for the receptor binding site. Now, an antagonist is actually going to bind to the receptor, but it's not going to lead to the same response. It's actually going to block the response. An example of an antagonist to our epinephrine is a drug that you would take known as a beta blocker. Why are beta blockers antagonists to epinephrine? When epinephrine binds, it can cause your heart rhythm to increase. So it basically speeds up your heart contraction. So in patients that need to slow down their heart rate, they would take a drug known as beta blockers. And those beta blockers, they would basically bind to specific receptors on the surface of your heart cell, and they are antagonists to your epinephrine. So they are actually going to slow down your heart rate. This image right here shows that you can have one ligand, and this one ligand can lead to different responses. How come one ligand can lead to different responses? It has to do with the type of receptor that ligand is binding to. So epinephrine has the ability to bind to two different types of receptors, alpha and beta 2. Alpha and beta 2 are a type of receptors known as adrenergic receptors. And the reason why we call them adrenergic because another name for epinephrine is adrenaline. Now, alpha receptors, you can find them on the surface of the blood vessels within your intestines. So when epinephrine binds to an alpha receptor, what happens is that it's going to constrict those vessels. Beta 2 receptors, which are usually found on the surface of the blood vessels found in your skeletal muscles. When epinephrine binds to a beta 2 receptor, what happens is it's going to cause your blood vessels to dilate. So one more time, epinephrine can either cause your vessels to constrict or it can lead your vessels to dilate. But what determines whether your vessels are going to constrict and whether your vessels are going to dilate is the receptor that epinephrine binds to. Now, an agonist to epinephrine is norepinephrine. So norepinephrine also has the ability to bind to an alpha receptor and a beta 2 receptor. But usually if you have both around, epinephrine and norepinephrine, norepinephrine would be favored to bind to an alpha receptor, and epinephrine would favor to bind to a beta 2 receptor, because beta 2 receptors have higher affinity for epinephrine and alpha receptors have a higher affinity for norepinephrine. Let's say that a signal molecule or a ligand is present in high concentration amounts in the body for a period of time. Well, what's going to happen is initially your cells are going to create an enhanced response due to having that much ligands within your cells. But then the cell would want to bring the response back to normal levels. So what's going to happen is downregulation. Downregulation, when it takes place, the cell goes ahead and removes some of the receptors on its surface. So if we have the cell right here with its receptors where the ligands would come and bind, the cell would go through endocytosis, where the cell is going to dip inwards, where one of the receptors or a few of the receptors would actually get pushed into the cell. and so the cell would have less receptors on its surface. By decreasing the number of receptors on the surface of the cell, we decrease the amount of ligands that would bind and therefore decrease the response. But that's why some individuals would start developing drug tolerance, where the response to a given dose starts to decrease, even though they are still taking the same amount of the drug. And that's because their cells are starting to go through downregulation and decreasing the amount of receptors on the cells. Now, the opposite takes place. So if the concentration of the ligand decreases and the cell wants to increase the response, what would happen is the cell would actually go through something known as upregulation, where the target cell that has those receptors on it would go ahead and take some of the receptors within itself and would actually push it up and put it on its receptor. So by increasing the amount of receptors, you can bind to more ligands and therefore increase the cell response.